Okay friends, now we are going to talk about a very important part of a bacterial cell. Probably it is a flagella, and I personally like this flagellar motor as a motor because this is, according to me, is one of the most remarkable motor which we can find in the world. And the design of this motor is really complex, and that tells us about the various magic of evolution. So this is simply just a fantastic example of evolution, and how a simple structure can be evolved to make such a complex structure like a flagella. We'll look in a moment. Okay, now what is this bacterial flagella? Bacterial flagella is simply an extension of the cellular proteins from the bacterial cell into the outside of a bacterial cell. It is it is a very long, it is many times longer than a bacterial cell on its own, and it helps the bacteria to propagate into the environment. It helps the bacteria to tumble. It helps the bacteria to move from one place to another place according to the response for chemicals. So this bacterial flagella can be rotated as a result of chemotaxis, which helps the bacterial cells to move from one place to another. And we not only can see or find this flagella in case of only bacterial cells, but we can also find this kind of flagella in case of eukaryotic cells too. In case of eukaryotic sperm, we know that sperms of mammals, we can find this kind of flagella. Not only mammals, but eukaryotic in eukaryotic higher order organisms, the sperm have the flagella, and this flagella help for those sperms to move and go to the egg and finally meet with them. Now here we in this discussion, in this in this presentation, I am going to talk about the structure of flagella. And first I am going to talk about some basic properties of flagella, then where we can find this flagella, and according to the flagella how we can divide bacterial system. Then we'll talk about the structure and assembly of flagellal subunits and how flagella actually functions, and we have some animations to say. And then we'll describe how bacterial flagella responds to the chemicals using the chemotaxis system. And then we'll talk about the evolution of bacterial flagella and the probable way of how a bacterial flagella can be evolved. Now let's begin. Okay, so let me take a color here, and now let's, okay. Now this is an example of this is a picture which is giving an example. So if this is a bacterial cell, then we can find there are lots of flagellas are coming out of a bacteria. So I have some thing to say before start this flagellar discussion, because we can find this flagella not only in bacteria, but we can find this flagella in archaea as well as in eukarya. So the three domains what is existing now on planet, all having this flagella. So this flagellar evolution must have a very important role for maintaining the very important properties of cell, like motility and other parts, or sometimes reproduction, when we come to the flagella of eukaryotes. Now as we can see here, the flagella, not only we can find one flagella at a cell. So the flagellar presence in a cells not depend on a particular cell type. So it can be present in one flagella in one cell. We can have more than one flagella in a cell. We can have flagella's arrangement different from different sides. So if you think this is a bacterial cell, so this is a cell like this is not a round shape, this is a oval shaped cell, and this cell, as you can see, flagella can come out from the side places as well as they can come from this polar places. Okay, so there are many different arrangement, various different arrangement that this flagella can do, as you can see here in this particular slide. Then we can find two different locations for flagellar origin or flagellar assembly. One is the polar assembly, the flagella, in which case the flagella will start from assembling from the polar side of a bacterial cell, and another one is a lateral flagella. In this case, those flagella can come from this lateral part of the lateral surface of bacterial cell. So that's why, depending on the origin of bacterial flagella, we can divide the flagellar group into this. Now, as we can see here, according to the emergence of bacterial flagella, we can divide, we can have a nomenclature upon those bacterial cells. Now you can see 4 different scenarios of bacterial flagellar assembly in different regions of bacterial flagella. As we can see in first place, in the picture A, we can have a flagella is coming out from only one polar region of a bacterial cell. In this case, we call this kind of bacteria a monotrichous flagella. Okay, this type of flagella are called the monotrichous flagella, or monotrichous bacteria which is containing the flagella at the monotrichous position. That means mono means one, so we have of flagella only one of these polar regions. Example of this kind of bacteria is Vibrio cholerae, which is the causative agent of cholera. And also Pseudomonas aeruginosa, which is a causative agent of cystic fibrosis. Now if we look at the second picture, which is the picture B, we can find this arrangement of flagella from one pole, but not one flagella. We can see many of the flagella. So instead of one flagella, which you can find in monotrichous, we have many flagellas. So as the name suggests, in this case also we name it lophotrichous. So trichous means it is coming out from one pole, and lopho means there are a lot of different flagella is coming out, not one. So example of this is Pseudomonas fluorescens. So this Pseudomonas fluorescens is a very important bacteria which it can generate fluorescence. Okay, it can generate bioluminescence, for example. Now in the third case, we are having flagella which is coming out from both of the poles of this bacterial cell. And as we know, instead of in the previous two pictures we are having flagella come out from only one pole, but in this case the flagella is coming out from both of the poles. In this case we are called them the amphitrichous. Amphi means both side, and trichous means it's coming out from the poles. So amphitrichous means it's coming one flagella at a time, is coming out from both of the ends. We call it the amphitrichous. The example of this kind of bacterium is Aquaspirillum serpens. Okay, so this case only one side flagellum operates at a time. So there is a question that, as we know, this flagella is helping the bacteria to go and move around. So as we know, if the flagella is being tumbling up and down direction like this, so let me erase the color. So this is the problem actually. So here is this. So it can tumble like this or go and run like this. That it helps the bacteria to move forward. But what happens if we have this flagella in both the direction, because they will cancel each other out? But the answer is, in this type of flagella, in the type of this amphitrichous flagella, only one flagella is active at a time, at a particular time. Then after that the other flagella will be active. Now in the fourth scenario, we are having flagella coming out from all the places of a bacterial cell, in both the ends and also both the lateral regions. So we are having polar flagella along with the lateral flagella in this picture. So we call it the peritrichous flagella. That means flagella is coming out from the periphery, or the whole surface of the bacterial cell. So one example of this is a very common bacteria, as we know, is E. coli or Escherichia coli, and also Salmonella typhi, which is a causative agent of typhoid. Now these are the examples of how we can find flagella, and it states us some properties about flagella, that the flagella is made up with some cellular proteins which can be assembled from the inward of the cell and going outside of the cell. Second thing, flagella, there is no particular regions for the flagella assembly inside the cell. Flagella can be assembled at the end or polar sides of a bacterial cell, as well as we can find flagella assembling at the periphery of a bacterial cell. Okay. Now here is another type of example, which is the example of organized flagella or peritrichous flagella, as we can see in this case. Organized flagella, for example, in this picture, this is a bacterial cell. This is an example of Selenomonas species. So it's a bacteria of Selenomonas, and in case of the Selenomonas species, what we can find is they make flagella from the periphery. So we are having this lateral flagellas producing. Now what they are doing in this case, they are arranging these lateral flagellas in different they arrange it in just coiled manner in like this. So they are producing this flagella, lots of this flagella from the periphery, and they organize those flagellas together. They organize them in this circular bundle like structure to make something very stacking structure outside their cell. They organize all their flagellas into a particular region. They're focusing all the flagellas into a particular region. By doing this, what they can do, they can maintain this characteristic shape of them. As you can see, at the site of their flagellar arrangement, there is a curvature they can create. So this curvature creation and their movement can easily be done using this kind of flagellar assembly, which are called indeed fascicles. So in case of this Selenomonas species, we can find the arrangement of their flagella is like fascicle. Okay. Now we have to see another example. In this example, what we can see, the flagella of spirochetes. The flagella of spirochetes are having some unusual characteristic than the other types of flagellas, as we can see in this picture. Then this kind of flagella, as we can find normally in the previous picture, if I go back, all these flagellas we are starting from inside the cell and going outside the cell. So most of the part of the flagella is being exposed to the outside of the cell, so the environment where the cell lives in. But in this case, in case of spirochete flagellas, what we can see, the flagella is placed inside the cell. So what we are having a cell. So we are having a cell here. For example, this is the protoplasmic cylinder. So we have a protoplasm here, and outside this protoplasm, what we are having this cell membrane. So it's the first cell membrane, and then we have outer cell layer. So we have two cell layer. One is the cell membrane, then we are having the outer sheath. So in between this cell membrane and outer sheath, what we call a periplasmic space. So this part is called a whole periplasmic space, this yellow colored part in this picture. So this periplasmic space, we can see in this periplasmic space they start to make their flagella. So what they are doing in this case, instead of producing flagella, so if this is a cross section of spirochete, so we have a long fibrillar structure of spirochete. If you make a cross section of it, we can find here. So normal cases, what happens, the flagella assembly begins something like this, from end of like that, the flagella arranged like that. But in this case, instead of this vertical arrangement of flagella, we are seeing the lateral arrangement of flagella, as we can look at this picture. So instead of flagella emerging like this, the flagella is emerging in this lateral line like that. Okay, so in case of the spirochetes, and not only we can find this lateral assembly of flagella, but also we can see the flagella is embedded inside the bacterial cell, inside the cell, where we can find inside the periplasmic space. Okay, and this flagella has a specific name again. They are called axial filament because they function like filament, they looks like filament. So they arrange themselves in such a way like to make this cell of spirochete behave like a screw. So they can transfer, they can move. When they can move, we can feel it resembles the movement of a screw. Okay, so it helps them to move faster, and it helps them to move really fast and forward. Okay, as we can see in this picture. So that's why these axial filaments are important for spirochetes. So as we can see, in all these different varieties of flagellar arrangement which are unconventional, must have a property, must have a unique property which supports which functions some important job, which generally do some important job for that bacterial species. So that's why they change their change the type of flagella from conventional to the unconventional one. Now let us talk about the structure of a bacterial flagella. As you can see in this picture, a bacterial flagella is really complex. Yes, it is a very complex structure indeed. As you can see here, we are having this peptidoglycan layer, which is the middle part, and we are having this outer membrane, and we are having the inner membrane. So this is the peptidoglycan layer. I am doing this peptidoglycan layer just very bad meshy structure like that. Now what happens, so from here on, this is the membrane, inner membrane. From here on, we are having the cytoplasm here, we are having the outside of the cell here. Okay, so we can see very tiny part or very little part of the flagella is being produced only inside the cytoplasm, but most of the part is just expressed outside the cell, as we can see. And this inside part, as we can see in every type of motors, as we know, the flagella is a motor. So just keep this thing in your mind, that this flagella is a very efficient motor indeed. So as it is a motor, then it must have to have a rotary arm, and it has to have a stable arm. So it has to have two things, one rotor unit, another is a stator unit, and that is exactly happening in case of flagella too. So it also have a stator unit which is embedded inside, which is embedded in this peptidoglycan layer, because this is a very strong and rigid layer which is holding the structure of whole bacteria. And it also have this attached to this outer membrane when in case of gram negative bacteria. So these are actually the stators which you can find in bacterial flagella. But in other things we have to produce a rotor which initiates the rotation of flagella, and in this case this initiation of rotation of flagella rotor is placed inside the cytoplasm. So the rotor of flagella is embedded in the cytoplasm as well as in the inner membrane. So this part, which is made up with the protein F, most of the part is the rotor of bacterial flagella, and this rotor of bacterial flagella is called MS ring. Okay, we'll see in a moment a whole animation about the structure, but this is the basics. It has to have a rotor, it has to have a stator. The stator is embedded in the peptidoglycan layer and sometimes in the outer membrane in case of gram negative bacteria. Gram positives are lacking that outer membrane, so in those cases they are not embedded in outer membrane. But the rotor of bacterial flagella is always placed on the inner membrane of bacteria as well as in the cytoplasmic phase. Now they produce the protein units one by one from here on. So as you can see, the protein which make the flagella is called the flagellin. So there are lots of different types of flagellin proteins. There are these are called the flea proteins, so flea F, flea M, flea N, and all these different varieties of proteins. There are thousands types of proteins are there which is being produced inside a cell to assemble to make the flagella. So they produces all these proteins together. As you can see here, they produces, and they will move from here on, from this below portion, from throughout this basal body, throughout this rotor, finally go through this structure, and finally it will go and assemble at the top. So the actual assembly process begins from the cytoplasm, and the protein molecules will go and stabilize. So again, second step of molecules go and they arrange, third molecules go and arrange. So the arrangement begins from the below to the above orientation. Okay, so the below to the above orientation. That's why I need to say this. Now let us look at an animation where we can find the whole structure and assembly process in detail. Okay.
Many species of bacteria propel themselves through their environment by spinning helical motorized flagella. Rhodobacter cells have one flagellum each, whereas E. coli cells have multiple flagella that rotate in bundles. Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell surface, make up the three parts of the flagellum. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into two major parts: the stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates. The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between two types of proteins, called mote A and mote B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in mote B functions in proton conductance. Each stator contains two mote B proteins, and therefore also contains two of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the mote B proteins. This binding causes a conformational change in mote A proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the two protons are released into the cytoplasm. The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of flagellar assembly have been determined. Flagella begin their assembly with structures in the inner membrane. 26 subunits of an integral membrane protein called Fle-F come together in the plasma membrane to form the MS ring. The Fle-G proteins assemble under the MS ring. Fle-G, along with Fle-M and Fle-N proteins, make up the rotor. Flagellar proteins destined to be part of the extracellular portion of the flagellum are exported from the cell by a flagellum-specific export pathway and assembled at the center. Mote A and Mote B form the stationary part of the flagellar motor, the stator. Both are integral membrane proteins, but Mote B is also anchored to the rigid peptidoglycan layer, keeping the stator proteins fixed in place. The subunits of the rod portion of the rotor move up through the hollow cylinder in the assembly, and assisted by cap proteins, build up the rod in a proximal to distal fashion.
Now in this case I must say one thing, that this cap protein, or as we can see, this is a protein which is a pentameric orientation. This is called a HAP complex or HAP. Most importantly, it is known as the HAP2 complex. This HAP2 complex has an orientation of pentameric, and this orientation are placed in such a way that these HAP proteins start to rotate on, and one by one the flagellin proteins which are being synthesized inside the cell, inside the cytoplasm, will start to come out and being attached to one by one via this circular arrangement, via this what you can say, this axial arrangement to make this helical structure. Okay.
Another set of rings, called L and P rings, are found in gram-negative bacteria such as E. coli. They penetrate the outer membrane, forming a bearing for the rod. As the rod cap is exposed outside the L ring, it dissociates and is replaced by a hook cap that guides the assembly of the hook proteins.
Now another thing I must say about these hook proteins, now as you can see, hook is a joint or joint region between this basal filament, the filament protein which we can see after that, and this basal proteins. So that's why the hook proteins have to be really strong, really flexible in nature, otherwise they cannot maintain the structure. As it is a joint for the outer part of the filamentous part of the flagella and the internal part of the flagella, it needs to rotate. So when the bacterial flagella starts to rotate, it has a force all time produced in these hook regions or hook protein regions. That's why this hook protein region has to have really flexible in nature, otherwise it can be broken down from this place. So how these hook proteins can be that much flexible? Due to the presence of their structural conformational shift that can be occurred due to rapid change. As we can see, these hook proteins, if we look at the structure of hook protein in much more detail, that we can find these hook proteins are arranged in different angular orientation with each other, and they can change this angle as when they need. That means, if suppose in this picture, as we can see, we can see arrangement. We can see, if I take the color, so as you can see in this picture, we have a bending like that. Now when the hook protein needs to bend this flagella in the opposite orientation, that means in this opposite orientation, the hook protein will start to put some of those proteins. As we can see in this picture, hook proteins which are placed in this region are made up with are slightly straightened in angle, but hook proteins placed in this are tilted in angle. When we have a situation when we have to bend this flagella in the opposite orientation, then this protein arrangement will be changed. So the proteins which is bound in this state will start to have a straightened conformation then, and the proteins which are placed here here will change their angle with each other to make to make this hook bend in the opposite orientation. That's why the arrangement of these hook proteins along with their own structure will help them to bend this filament along with the hook, and that is really necessary for the movement of flagella. If this flagella becomes rigid, then the purpose of having flagella will be failed, right? Okay.
After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and future filaments. Finally, yet another cap is built, and filament proteins assemble. Like the rod and hook proteins, they travel through the hollow channel inside the filament to reach the distal end. The cap rotates, which causes the subunits to build in a helical fashion. A complete filament can consist of 20,000 to 30,000 subunits.
That's how the flagella is being formed. As you can see, the flagella length can be several times higher than the bacterial cell. Okay, now we are going to talk about another portion of our discussion, which is the chemotaxis machinery. Now it is very important for flagella to move according to the chemical responses, because the bacteria have to move from one place to another place, and the importance of having flagella or making flagella like a motor is nothing but to maintain the movement of bacterial cell. So this is our actual goal, is to rotate the flagella and finally move the cell from one place to another place. So that is called the chemotaxis, as this movement is based on the signal which is coming out by the type of chemicals. Now, as we can see in this picture, is the illustration of how chemotaxis machinery is being occurred. So you can see this is a complex picture, but don't look at in this way. So just think, first of all, this chemotaxis machinery of bacterial flagella depends on, or divided into two different parts. One part is the controlling or the movement of flagella, and second part is the sense adaptation. So it is made up with these two parts. One is the movement of flagella, and the second one is the sense adaptation. So let's talk about the movement of flagella first. So this is the movement of flagella, and second one is the sense adaptation. So let's talk about this movement of flagella. So what happens when a signal comes in, which is here in this case aspartate as a signal, which is come inside the inner membrane of flagella, where the flagella start to assembly, where the rotor of the flagella is placed. As we can see, there are different arrangement of proteins are there for the signaling purposes. We have the tar proteins, which is a dimer condition of the protein, so it's a dimer of tar proteins, and we have this W, which is called CHEW. By the way, in all this case, you can see Y, Z, B, R, all these proteins have the abbreviation of CHE, or chemical protein R. So CHE protein. So these are the CHEW protein, and we have a dimer of CHEA attached to this. So we have a complex of tar, CHEW, and dimer of CHEA proteins attached, which is a primary receptor for the aspartate signal. Now when they receive the signal, what happens? In very first situations, when they receive this signal in normal time, now first thing I must tell you before beginning all this thing is that a bacterial flagella never stop working. So as it is a motor, something it have to do all the time. That means either it have to rotate or it have to run. So it is rotating in such a way that it helps the bacterial cell to run. That is happening very few times when they need to chase, when they need to take food. But in other cases, for maintaining the cell into the environment, the bacterial flagellum has to rotate in up and down motion, which is called the tumbling of bacterial flagella. And this tumbling is important, and without this bacterial flagella tumbling, bacteria cannot maintain itself in the environment they live in, normally especially in the environment when they have the watery content, right? So in this case, most of the time they are making the tumbling all the time, in fact, but very rare occasions they have to move from one place to another place. In those situations they will run. In those situations they will receive the signal and run. Now what happens in this case, when we produce the normal situations, as we can see, when the aspartate signal comes in, and this CHUY molecule is there, the CHUY is phosphorylated. When CHUY protein is phosphorylated, it becomes active, and it will go attached to the motor. And as soon as it attaches to the motor will start functioning the run procedure. It will only tumble, so it will only maintain the balance. So at that situation, okay, and there is a protein inside the cell which is called CHZ, which converts this which dephosphorylates this CHUY protein, and thus set back this motor from their tumbling state towards the run state, as we know. Now what happened in general situation, as we can look in this picture, when we have aspartate coming in, then what happens is aspartate attached to that, and it activates this total complex of CHE, W and TAR. It finally leads to activation of CHUY, and CHUY is dephosphorylated by CHZ, and finally what happens, the motor will start to run really fast. So this is the signal, as we can say. So we have a run and we have a tumbling. So run and tumbling motion. Normal situation we always have this tumbling motion. I am extremely sorry for my extremely bad handwriting, but whatever, this tumbling motion is going on all the time. But what happens whenever we have a signal coming out from the outer place, and which as a result of CHZ, which is dephosphorylates CHUY, finally turns on the state of the motor, which helps the motor to run really fast. Now this is the way how we can control. In general situations, when we have CHUY phosphorylated bind to the motor, it stabilizes. So if it binds to the motor, it stabilizes, only tumbling, okay. Or if CHUY go and work to the motor, it helps this motor to run. So that is the situation. When you have lots of aspartate as a signaling, this signaling is happening. When there is less aspartate, that means there are less ZD to convert. So CHUY and attached to this CHUY phosphorylated attached to the motor, and that in turn helps the motor to tumble only, not for the run. Now we have to control this sensing of the chemical attachment. Now how they can sense this chemical attachment? We have a internal intracellular chemical process, as we can see here. We have S-adenosyl methionine, and they convert this S-adenosyl methionine into methyl and S-adenosyl homocysteine by CHER protein, and by doing this they produces lots of the CH3 or methyl groups. And what is the importance of methyl group? When you have lots of methyl group present inside the cell, it will attach to the TAR complex and make this TAR complex extremely reactive. When they make this TAR complex extremely reactive, as a result of that, a small amount of aspartate can carry on the signaling and can help to run that. So all the time, if this methyl group bind to the TAR complex all the time, then the cell will remain in the state of the running state, and this flagella will move really faster. It helps the cell to run properly, okay. But that is not necessary all the time, because the cell do not always have to chase something. The cell do not have to go for eating some food all the time. So it will extremely loss of energy. As we know, to run this motor we need ATP, as we can see here. So that will be the loss of energy all the time if it is activated all the time in the super active form. That means if this TAR complex always bound to the methyl group, so we need some way to convert this methyl group and take this methyl group out from this TAR complex. And how can the cell manage this? The cell regulates this situation by the backward process, which is mediated by this another protein which is called CHB. As the CHB, it is as we can see, this is called adaptation of this signaling. Now CHB is when this CHB is phosphorylated, then the phosphorylation of CHB causes the uptake of methyl group from the tar complex and to make another alcohol, which is CH3OH, which is a methanol, the production of methanol by this conversion. And that is why and by using the CHEB, which helps to uptake the CH3 from tar and makes methanol, helps to block the cell to be go all the time at a state of super reactive. So it prevents the cell to move on to the super reactive state by cleaving this methyl group out from the star and making methanol. Okay. Now this CHEB, the story of CHEB is very simple. It is always it's an autophosphorylation event going on between the CHEB and CHEB phosphorylated. So it is being phosphorylated then dephosphorylated all the time, and when it is being phosphorylated, then it grab the methyl group from the tar and make methanol, but then it is again dephosphorylated in its original form. So that's how they balance and they regulates the signaling or the sense of signaling from the outer environment. Okay, and that is really important, and that is nothing to do with the movement of motor, but this is have to do with the economy of using ATP, because ATP is a very important ingredient inside the cell. As we know, ATP is the energy currency for a cell. So the sensing along with this motor movement is important to maintain the balance between the chemotaxis inside a bacterial cell. Okay, now we'll look an animation which shows that. Now if we see this animation, we can find. Now here we have aspartic acid which is coming as a signal. We have tar complex CHEW and CHEA complex. As it is binding, it is the CHEA molecules are coming and bound to that. So we have make a complex of motor, and it is in turn activate the CHEY and attach the phosphate group into the CHEY to make the CHEY phosphorylated, which is the active form of CHEY, and it is going on, and it is go and attach with the flagella inside the cytoplasm, which stabilizes the SW state. That means the inhibition of the rotation. It doesn't mean the inhibition of rotation, but it means the inhibition of run. Now if CHZ comes in and degrade the phosphate, it will stabilize the opposite state, which is SSW, which state means the running of the flagella. Now what happens, CHER take the SAM and convert it into SAH and produce the methyl. As you can see, when the methyl group is produced and attached to the tar complex, it will activate this tar complex really fine. So it is activating the cell. It is locking the cell into a very hyper reactive state. So this state will produce more and more CHEY phosphorylated, and CHZ comes in and dephosphorylates them. That means flagella will be put on a very running situation, or the situation when the flagella will move really faster to run this bacterial cell. But not always we need that, because if we always have to make the cell to run, then it will be not necessary for us. It will be very loss of energy all the time when we start to run our motor, right? So that's why we need to have an adaptation complex in this case, and that is provided by the CHEB. As the CHEB always try to be phosphorylated and dephosphorylated, the phosphorylated form of CHEB comes in, it will grab the CH3 and make methanol, and this methanol production in turn is gonna block the hyperactivation of tar complexes. And as you can see, these autophosphorylation events are also being controlled by that. So that's how this whole chemotaxis as a combination of motor movement as well as the sense adaptation helps the bacterial cells to move on according to their environment sensing, as well as tumbling, as well as maintaining their balance in the environment and the run process. Okay, so now in this part we will talk about the evolution of bacterial flagella. Okay, and how a bacterial flagella probably can be we can say evolved from the previous form to this particular form. We'll see. So we have a background music here, and you have fewer narrations in this place, but still it will help you to notice that.